Okay, so let's go. I'll start, Johanna. I'll speak some things in Portuguese, so
Go ahead.
So everyone can understand us, and then I'll signal for you to start, ok?
Okay.
Então, bom dia, gente. Eu sou o Paulo. Eu estou aqui com a Johanna, que vai dar uma palestra para a gente, né, sobre um transporte de água que a gente tem em algumas plantas, algo bem incomum que a gente não vê geralmente, né, mas eu não vou falar muito para não tirar o suspense. Então, a Johanna, ela fez a graduação lá na Universidade Nacional da Colômbia, em Bogotá. Depois ela foi para os Estados Unidos, onde ela fez doutorado em Washington. E depois ela fez um pós-doutorado em São Luís. E imagino que o que ela vai apresentar para a gente é principalmente os resultados desse pós-doc. Então, thank you, Johanna. Thank you for coming to share your results with us. And you can start whenever you are ready.
Okay. Thank you, Paula, for the introduction, and thank you for inviting me. So I'm just gonna start sharing. Can you see right now? No. Yes? Can you see my screen?
Okay. Yeah, it's okay.
Okay.
So yeah, we can see.
Okay. Great. So this photo that I'm showing you here, I want you to just look around and check very carefully. This is a photo of a big leaf maple tree, and it's a photo that is viewed from the bottom. And as you can see, if you check very carefully, these are the legs of my field assistant. She's actually sitting on a branch of this tree. And what I want you to notice very carefully is that actually this tree is completely covered with mosses and plants. So as you notice, the trunk is completely covered, and here the branches are actually also covered with mosses. So this moss eventually, and all these plants that grow over the branch, accumulates and decompose and form something that we call canopy soil. And the canopy soil is actually the focus of my talk today. So the title of my talk today is Changing Directions, Tree Hydraulic Redistribution from Canopy Soil. This is a project that was done at the Plant Ecophysiology Lab and Plant Grow Lab at the University of Washington in Seattle. And it was directed by Dr. Liz van Volkenburgh and Dr. Soo-Hyung Kim. And I'm going to start talking a little bit about history. So for a while, for a long time, a lot of scientists, biologists, plant biologists, physicists, engineers, architects have a lot of interest to try to resolve questions about how actually water moves in the whole, in the planet. Basic and most important, they are actually focusing on how and trying to explain how water moves against gravity. And so for many years also, organisms have developed different adaptations to cope with this challenge to move water against gravity. And probably you're wondering why I am showing you this giraffe here. So actually the giraffe is the tallest animal in the world, and they have developed these huge hearts that helps them to pump up the blood from their main body to their brains. And so the heart is the biggest heart in all the animals that actually just work as a pump. But what about trees? So here there's a sequoia tree, and sequoia trees are the tallest trees in the world. So if you imagine giraffe can reach around 5 to 6 meters tall, a sequoia tree can reach around 150, 124 meters tall. So it's actually 24 times taller than a giraffe. And also the trees and plants have to deal and cope with this problem, or that is moving water against gravity. So trees also move water from the bottom, from the soil, to the canopy. But actually trees don't have a heart or don't have pumps that help them to pump up or to push the water from the bottom to the canopy. So how do they actually do it? So the answer to this question are water potential gradients. So water potential gradients are symbolized with this. This is a Greek sign called psi, and then we have a subindex that is W. So water potential refers to water. And water potential refers to all the free energy that the water has to move from one side to the other. So when we talk about completely pure water that has all the freedom to move from one side to the other, we say that it has the highest water potential because it is completely free to move. So pure water will have the highest water potential, and it will be equal to 0 megapascals. So water potential is measured in units of megapascals. It's a pressure unit, and 0 will be the maximum. So anything that can difficult the movement of water from one side to the other, then this value of water potential will be lower than 0. Also, I want to tell you that water potential has different components. So here there is a component of solid potential that is referred to the osmolytes, or the amount of solids that are in the water. So more solids will difficult that movement of water from one side to the other. There is also a hydrostatic pressure potential that refers to the force of the water or the water molecules against all the cell walls. And usually we talk about this as the turgor pressure. And then we have the potential of the gravity. That is the force that exerts the gravity against the water molecules. And we have the matrix potential that refers to the surface tension. So it's actually those forces that attach the water molecules to the cell walls, and also the forces that attach the water molecules among themselves. So this is just to tell you that in the sum up of all these components, it's equal to this water potential at the end, but we're just going to focus in general to the whole water potential and not to each of these components. So if you see to the left, there's a diagram of how actually water moves within a tree. And then we know that water moves from very high water potential to a low water potential. So if we move across the tree to the body of the tree, we're going to see that actually at the end, there's going to be the lowest water potential here in the atmosphere. That is why it lets make the water to move from high to the lowest. So if we add numbers to the tree, so here you can see that soil is around minus 0.3 megapascals. And then if we go up, roots will have a minus 0.6, so it's a lower water potential. And then stems will be minus 0.7, leaves will be around minus 0.8. And then this is a very important here step. In the atmosphere, you see in United is around minus 70 megapascals. So this huge difference between the leaf and the atmosphere, so it's between minus 0.8 to minus 70, this huge gradient of water potential is actually what exerts that force and generates that very negative tension that pulls up the water from the soil to the leaf, where the evapotranspiration occurs. And so as we notice, so then all the water molecules are attached to themselves. They form this water column that moves based on that moves in direction based on the order of these water potential gradients. So it has to occur this very, very huge difference between the leaf and the atmospheres to be able to pull up the water in this direction. But then, if the water depends on the order of these water potential gradients, what would happen if these water potential gradients change? How actually will be the movement of water within the tree? And before answering this question, I'm just going to talk a little bit about myself. So I was born in Colombia, just here. In Colombia is in South America. Third of the country is actually part of the Amazon rainforest, and I guess you're very familiar with the Amazon rainforest. And this is just a typical photo of the tropical rainforest, as you can see here. And most of it is actually green. It's kind of a little bit difficult to distinguish between the trunks and the branches because all of them are actually covered with epiphytes. And we know epiphytes are all these plants that can grow over the branches or over the trunk of the tree their whole lifespan without touching the ground. And so most of these trees are completely covered with epiphytes and plants. In my case, I was very lucky to be able to do my research in this other type of rainforest. This is a temperate rainforest, and it's located at the Olympic Peninsula, just at the Pacific Northwest of the United States. And as you can see, it's also a rainforest, but it's easier to localize or identify the trees, but also most of the trees are completely covered with epiphytes. So big difference, of course, because of their location. And this is a temperate rainforest. This is a tropical rainforest. So in the tropical rainforest, it is more predominant bromeliads, orchids. In the case of the temperate rainforest, it's more predominant the mosses and actually ferns that actually cover all this part of the trunk and the branches. And so eventually they accumulate, and as I just told you at the beginning, they accumulate. All that tissue also accumulates over the branches and form organic matter. Also, in these parts of the these habitats, also there are insects, there are microorganisms, there are invertebrates that live there, but also they become some eventually organic matter that accumulates over the branches of the trees. And then they start forming a huge layer of biomass over those branches. And this huge layer of biomass becomes eventually something that we call canopy soil. And we call it canopy soil because it's actually very similar to the soil underground. Their structure and composition is actually very similar to the soil underground. And so it can reach, actually this huge biomass can reach around 30 to 40 centimeters of thickness, just right above, just on the branches. And so we're going to talk a little bit about this canopy soil. So just again, let's see this our same diagram at the beginning. And then this canopy soil is actually located here on the branches. And it could be also, because I told you it's made of organic matter, it's made of a huge amount of mosses. It also could be a potential reservoir of water and nutrients there. A lot of different studies about canopy soil. So this is a real picture of canopy soil. They have seen actually that the canopy soil is very similar in composition and chemically to the soil underground. So carbon and nitrogen ratios are very similar. And also it's very important to know that it has a large amount of biomass within the same forest. So actually the canopy soil, has a very important and significant role in the whole cycling of water and nutrients in the same ecosystem, in the ecosystem, and also are the microscopes or the habitat for different microorganisms, insects, and invertebrates. So it has a different function and significant function inside the ecosystem. So additional to that, so here they have found actually, so this is just a cross section of a branch here. And here you can see again all these epiphytes and plants that are growing over the branch. They have found, and then they decompose and they form what I told you that is the canopy soil. But they have found actually that there are also roots underneath this canopy soil. And these roots, we call them canopy roots. These roots are not from the epiphytes by itself. These roots are actually from the host tree. So these roots are actually growing from the branches, from the host tree, underneath that canopy soil. And so we have found and we have seen these roots. And so you can see here in real, so we just remove that canopy soil layer, and then the arrows are showing you the one of the real roots here. So I just zoom in here. These actual roots can reach around 10 to 12 centimeters of diameter, so very similar to the roots underground. So now we know also these canopy roots are also adventitious roots, and all adventitious roots, so we are very familiar with the principal root system, but adventitious roots are all those type of roots that grow in different and abnormal places in the tree. So it could be on the trunks or the leaves or on the branches. And several species have developed adventitious roots depending on the conditions or stress conditions, just in order to be able to find other substrates. So we can see that corn, pandanus, and different species have developed adventitious roots that if they have a substrate, they can be functional and they can uptake water or nutrients to help the trees. So now, in summary, I explained to you that water moves in this direction that's what we learn, and based on the water potential gradients from high water potential to low water potential gradient. But now we have this new system that is in this movement that are the canopy soils. And canopy soils, as I just told you, they could be potential reservoirs of water and nutrients. And so if there is water in the canopy soil, then also we saw that actually there are roots that probably are functional or potentially functional that can uptake water from those canopy soils. And so if we have a new reservoir of water on the canopy, then what would be then the direction of or movement of water within the tree? Because now all these water potential gradients probably will change. So it could be that probably the water is moving bidirectionally. So it could be that it's moving from the bottom to the top and from the top to the bottom simultaneously. Or it could be that there's only reverse flow if the conditions, probably if the soil it's drier than the atmosphere, so probably makes the water moves from the canopy to the bottom. So just as a reverse flow. So here's my question. So as I explained, so we know the movement of water from the bottom to the top, but now we have canopy soil that probably is changing or affecting the hydraulic redistribution of that water, not only in just one direction or probably at the canopy level. So I set up this new hypothesis for based on this problem. So this is the main hypothesis is that actually those canopy soils has an impact on the hydraulic redistribution. So if you notice here on the left, this is just the normal movement without canopy. And then what we are expecting is actually if there's canopy roots or canopy soil, probably the redistribution will be different. But not only the canopy soil will have an impact on the hydraulic redistribution, but also we wanted to know how actually will be the plant performance of, or how plants will respond if they have canopy soils under drought conditions. So probably under drought conditions, the canopy soils could be just an extra source of water that helps to alleviate the drought conditions of the tree. So to answer this hypothesis, we split this work in three different parts. This is me looking how I was going to deal with this problem. And so this is just a big leaf maple tree. We're going to start from the bottom here. So first, we wanted to know if actually the canopy roots were how anatomically how different are from the roots underground. If they were different or if they are similar, or if they, and in that way, how functional they are. Then we have a second part, that is how if these roots are able to uptake some water, and then if actually that water is redistributed within the tree. And finally, we wanted to know if actually this water helps in or impact the physiological performance under drought stress. So I'm going to start explaining principal methodology that we use and we apply for this research. So we did all part of our research at the Center for Urban Horticulture and at the University of Washington in Seattle. And we use poplar trees initially because they are very easy to propagate. We use poplar trees that were in the nursery. So these were the poplar trees that we used for some of the experiments. And we also propagate by cuttings in the greenhouse the same poplar trees, but just to do greenhouse experiments with controlled conditions. And then we also, as I show you, we did some, collected some samples on the Olympic Peninsula, but actually with big leaf maple trees that are Acer macrophyllum. And in this case, we, because they have canopy soils naturally and canopy roots naturally. So, yeah. And I'm going to explain now for the poplar trees. They don't develop naturally canopy roots, but we use this very common technique of propagation, the air layering technique, just to develop those canopy roots. So if you're not familiar with that technique, I'm just going to explain it very briefly in few steps. So first, you peel the branch, remove part of the, just peel the branch, and then add some auxins or growth hormones, and then you cover it with peat moss, so just to simulate the mosses or canopy soil in the forest. And then we cover it with plastic wrap, and then cover it with aluminum foil just to simulate dark. And so it's kind of like a fake soil there. And after a month, we got our canopy roots. So with these canopy roots, we were able to do some experiments inside the greenhouse and also at the nursery, easily done in the field. Okay, so let's start with the first question. My first question was, how different are canopy roots versus terrestrial roots anatomically? Are they actually functional? So for this experiment, my first hypothesis is that actually they are not. There are not anatomical differences between the canopy and terrestrial roots, and therefore, for me, they will be functional. So we just, based on their anatomic structures, we assume that if they have the basic structures, we assume that they are actually functional. And so for this experiment, we collected greenhouse canopy roots. We collected roots from canopy roots from the nursery, from the poplars outside. And then we collected also canopy roots in the field. And this is one of my field assistants. She's very happy because she grabbed her first canopy roots, and maybe you can see it here, but I highlighted. This is actually half of her body big. So we collected all these canopy roots, and we compared them with terrestrial roots that were collected from the same trees. Okay, so the first results that we saw, so first, before showing you results, I'm just going to show you here a diagram of a simple root. And this is roots are divided or classified by different, based on their branching. So the main root will be a first order root, and then the first branch of this root will be a second order, and the branch of this branch will be a third order branch, third order root. So in this case, we always collected, and in this study, we collected most of the second order roots. Sorry. Another thing that I want to explain is all different developmental stages. So close to the tips, we're going to see early development. So they are the youngest parts of the root. And then closer to the main root, or closer to the first order root, will be more mature, or more, you will see more mature developmental stages. So the first results that we have were just only visually observations of the canopy roots and the terrestrial roots. And then actually we didn't see very big difference between them. except that terrestrial roots actually are more woody, have secondary growth, and have woody tissue in the main roots. But besides that, most of them, they look very similar. So we expect probably these woody tissues only because of the age of the roots. Probably underground roots were older than the canopy roots. That also makes more sense. But besides that, we didn't see any big difference between them. Now, as I mentioned, we just collected a second order root, and then we collected from the microscopy observations were done first of the early stages of just the tip of the roots. And to explain the next results, I'm just going to show you this diagram of a cross-section of a root. And so the dashed blue line only represents the movement of water, or the pathway of the water to cross and to enter inside within the roots. So these are only root hairs. And then there are several cell walls that the water has to cross when enter into the roots. So the first wall will be the epidermis, that is here in green. And then it has to cross all this parenchyma tissue that are the cortex. And then finally the endodermis, where the Casparian strip is, and all the filtering and selection occurs. And then finally we find the vascular system here, that is the xylem and the phloem. So this is a very basic anatomical structure of a root. And then what we see, so I'm going to just start showing just the results. So these are canopy roots. This is just different resolutions of image. And these are terrestrial roots. So actually, what we see here is we could distinguish all the different basic structures. We could distinguish the epidermis, cortex, and the dermis. The main difference, again, is that actually the epidermis for the terrestrials are very thick, so showing probably that is just secondary growth. Same thing, we saw it in, we collected samples with other level of developmental stage. And again, we see exactly the same things. We saw all the different structures and very thick epidermis for the underground or terrestrial roots. So with these results, and then we calculate some differences about the diameters. So we notice we calculated the diameter of the root as a whole, the diameter of the stele, or the diameter of the xylem and the phloem, and the thickness of the endodermis. And actually here we found some differences, significant differences on the sizes of the root and the vascular system. But by itself, we found all the different structures. So if we go back to our hypothesis, there is no anatomical differences between canopy and terrestrial roots. Therefore, maybe they may be functional. We found all the structures. So all the roots present the main functional structures to work as functional roots. However, we found actually difference in like dimensional differences compared to terrestrial roots. But this could be only explained based on probably the age of the roots. But most of them, they had all the main structures to be functional. So now that we assume that those roots are functional, we wanted to see if actually those roots can uptake water from that canopy soil. And so to do this, we develop we have this hypothesis that actually these canopy roots can uptake water from the canopy soil. So we designed a dye tracking experiment. We use popular trees from the nursery, and these popular trees are hybrids between Populus trichocarpa and Populus deltoideum. We apply the air layering technique that I explained before. We develop all those canopy roots, and then we build a cone system. We immerse all those canopy roots and use dye. In this case, we use acifloxin. So we submerge all those roots in acifloxin, and then we wanted, and so here you can see the real pictures of those cones. And then what we wanted to see is actually the movement of the dye traces across the tree. So what we wanted to see, if there was water uptake, then we were going to be able to see dye traces around within the branch. So those were our results. So at the beginning, so this is just time 0, the first picture. And as you can see here, this is the canopy soil system. And we start peeling the branch every 15 minutes. And so at the beginning, we were not able to see anything. So there are no dye traces at all. But then over time, we were peeling and we were being able to see dye traces moving across the whole branch. So we peeled the whole branch, and we were able to see actually all the branch with dye traces moving towards the leaves. So the arrows are showing you the movement of the dye. So we only were able to see movement of that dye in one direction that day, though several times just towards the leaves at the beginning. And it would make sense since we did this experiment at noon, that is when the sun is in the maximum point, and so plants are transpiring a lot. So it makes sense that actually if there was a water uptake, that the water is going to move towards the leaves to evapotranspirate. But we were also interested if actually that dye could move towards the other direction, opposite direction. So in this day, we didn't see it, but the following day we went back, and then we actually saw dye moving towards the trunk. And even we peeled the trunk and we saw dye traces on the trunk. So actually, we also saw moving like dye traces moving in the opposite direction. So with this simple experiment, we repeated several times in several years, and we have a very consistent result that actually the dye was moving towards the distal leaves and also towards the trunk. So we could conclude that actually, yes, these canopy roots or the trees are taking water from that canopy soil. And actually that water is moving not only in just one direction, but also to the opposite direction. So probably bidirectional. So now we have a third question. So can this water move from the canopy soil and be redistributed toward other plant organs? So we already had some evidence that actually that water is moving in 2 directions, but we wanted actually to confirm that it can be redistributed to different parts. So to answer this question, we developed an experiment inside the greenhouse. And this was, we set up this hypothesis that actually water from the canopy soil can be redistributed to other organs within the tree. So we designed this experiment with 2-year-old poplar trees in the greenhouse, and we applied these 3 different treatments. There was a well water plan. So plants were completely well watered. So every other day we water them. We have trees under dry conditions. So the pots were completely dry. And then we have a DW treatment. There are plants that are completely dry at the pot level, but they have canopy soils. And then this canopy soil, so each tree has 3 different canopy soil systems that were connected with IV bags. And then we were administering water through the IV bags directly to the canopy soil. So just making sure that the pot was completely dry. And so these trees were only able to uptake water from those canopy soils. Okay, so in order to know if there was actually hydraulic redistribution, we applied 2 different methods. The first one was an isotopic analysis. So we did 2 different experiments in 2 different years. And we used 2 treatments in this case. We don't have the completely dry. We have the well water plants as control plants. And then we have the plants with the canopy soil. And then we use heavy water. And when we talk about heavy water as an isotope, so we know molecularly, we are all familiar with the molecularly of how water is. It's just 1 proton and 1 electron. And it's actually, that's the molecular form of the water. 99% of the water is not this form. But actually 1% of the water is also in their 2 other isotope forms. So we use in our case deuterium that it has a neutron plus that it makes it heavier. And then there is tritium that has 2 neutrons and is a radioactive isotope, so it's not stable compared to deuterium. So now that we have heavy water, we were going to be able also to track actually where that heavy water is going to move within the tree. So what we did is enrich those samples. So we have this is just a very simple diagram of where we add the water. So here we have the canopy soil, and we added heavy water or enriched this canopy soil with heavy water. And then we collected samples of different parts of the tree. We collected samples of the leaves that were on the same branch of the canopy soil, leaves from a neighbor branch. We collected samples of the branches, B1 and B2. We collected samples of the trunk, so upstream and downstream, that is T1 and T2. And then also we collected samples of the roots underground. So if the enriched water was able to be redistributed, we wanted to see if actually we can see that enriched water in all these different parts of the tree. And so we analyzed these samples at the Center for Stable Isotope of Biochemistry at UC Berkeley. And then now I'm going to show you our results here. So in 2000, these are our first 2017 results. And just to understand these results, I'm just going to explain this plot first. So again, just in the y-axis, we have R1 that represents the canopy soil. We have L1, L2 that represents the leaves of this branch, the leaves of a neighbor branch, and so on. So we have just different samples that we collected. On the x-axis, we have delta deuterium per mil. That is actually the units for isotopes or heavy water. And then each region has their own enrichment of tap water or normal water. So, or your tap water or the water that you drink has a specific value of isotopic enrichment. In the case of Seattle, that was the tap water that we were using for watering all our plants or the well water plants, the value was around -70 per mil. So it's this dashed line. So everything that was enriched should be above or bigger than -70. So it will be to the right side of the plot. Everything that is less enriched will be to the left side of that -70. Okay, and just remember again, so WW were the plants control and DW are the green plants that are with the canopy soil. So I'm just going to show you first the control plants. And this is what happened. We of course don't have a bar for R1 because there were no canopy soil, but then we noticed, so some of the samples are around -70, and that's what actually we were expecting because we were watering these plants with tap water, but some of them were enriched already. And if you notice, the parts that were enriched are actually L1, L2, that are the leaves. And we have R2, that are the roots underground. And so if you think about what is happening in the leaves is usually, well, in the leaves we know that transpiration occurs. And so naturally, all the water molecules that are lighter will be released faster than the heavier molecules. So by nature, by itself, there is a certain fractionation in the leaves that occurs and then makes them more enriched than all the other organs, and same thing happens in the roots underground. There is certain fractionation between heavy molecules and lighter molecules of water that actually makes this tissue more enriched than the other ones. And now if we compare with the with the canopy soil samples, that are the green bars here, you can see that most of them are actually enriched. And so this is a graph of a summary of the results of this panel. So I highlighted where we were able to see enrichment. So for the control plants, we were able to see enrichment on the leaves and also in the roots. I didn't put it here, but in the plants with the DW plants, we were able to see enrichment in all the different samples that we collected. In 2018, and as you can see here on the bottom, the difference were not as significant as 2017, but still we were able to see this enrichment in most of the samples, mostly at the canopy level. So if you can see a summary of these results here, again, we were able to see here enrichment in the leaves for the control plants, and actually for the roots a little bit less than in 2017. But actually for the canopy roots, we were only able to see enrichment at the canopy level. So just in one direction upstream, but downstream, we were not able to see any enrichment. So with these results, we could conclude that with this hypothesis, water from canopy soil can be redistributed to other organs within the tree. Yes, we actually saw that in both years, water could see, we see enrichment at the canopy level in both years. And even in 2017, we were able to see water coming from going downstream, so including to the roots underground here. So another, we also use other type of method, subflow measurement sensors, just to check actually the velocity or the direction of that water. And so the subflow sensors apply this method that is a heat ratio method. And this heat ratio method consists of, so this is just a trunk. So this gray box is a trunk. And then the heat ratio method consists of heater probes. So it's based on heat diffusion. So these are 3 probes. These are the sensors are made with 3 different probes. There are 2 probes that are temperature probes, and there is one in the middle that is a heater probe. And then based on the water movement, this heater probe is just going to release heat, and then that heat is going to be dissipated by water. And then all these temperature probes are based on the difference, we will know what will be the direction. So if the heat goes, if the heat goes up, so then the difference between the upstream temperature probe and the downstream temperature probe will be positive. But if the water is going down, so the heat will dissipate downstream, and so then the difference between the temperature probes will be negative, and then we will be able to know that actually that subflow is going down. So in our case, we use a similar method of the heat ratio method. The only big difference is that we applied new subflow sensors designed by Adjijon. In this case, this was the first time that we applied these sensors, and these were less invasive. So this is just, again, the trunk. In this case, these probes are completely inserted in the trunk. So there are needles that are inserted in the trunk. In this case, they were only just touching by contact, physical contact, the trunk. And so we set up all these probes in our trees in the greenhouse, and we set up them just right below the canopy soil, and we got these results. So before showing you the results of the subflow sensor, I'm going to show you how actually normally subflow, what is the dynamic of the subflow in a C3 and C4 plant. So normally, so here is a plot. So on the y-axis, you have subflow velocity, and the x-axis is just time of the day. So this is 12 a.m. and this is 6 a.m. And as you can see here, because it's before the sunrise, plants are not transpiring. So actually the subflow is very low. But then during the day after the sunrise, you can see actually that the subflow is very active. And then around noon, we see the maximum point of subflow activity. And then again, after in the sunset, so around 6 to 12, again, the subflow will tend to decrease. So that's how it's just the diurnal dynamic of the subflow. So now in our results, here, again, the y-axis is the velocity or the subflow velocity, and here is just the time hour of the day. So each panel here represents a day of the experiment. So this is day 6, day 7, day 8, 9, 10, so on. And then here we have this line, there is the 0 line. Everything above the 0 line will be normal subflow. Everything below that 0 line will be reversal flow. And so again, just remember, WW will be the well water plants that are controlled, and then we have the plants with canopy soil. And so I'm going to show you first the plants with canopy soil. This is what we saw. And so on day 6, you can see here on day 7 that actually the normal pattern going down, going up, going down, going up. But actually on day 8, we can see that it's actually below 0. Again, day 9, and then on day 10, we see a very low reversal flow. If we compare this with the control at the end of the experiment, you can notice that actually control plants have a normal dynamic of subflow. And then the red lines are representing when we saw actually that reverse flow that we were expecting. So this critically explained. So just remember, we start with our plants, all of them at the beginning were well watered before we set up the treatments. And so it's probably that these plants with the canopy soil, on day 6 and 7, probably they were still having some available water on the pot, and they were just acting as normal because they have more water available at the pot level. But by day 8 and 9, these pots are getting drier because we didn't water them at all at the pot level. And so they probably are starting up taking water from where they have more water available. It's at the canopy level. And by the end of the experiment, there is no water at the pot level. So we were able probably to see that these plants are just uptaking water from the canopy soil. So with this experiment, we again confirm what we saw in the isotopic experiments, that actually water was redistributed to different parts of different organs of the tree. And then we also, we observe actually reversal flow in these trees, supporting that water redistribution. Okay, so we finished the first part, and we know now that actually the roots are functional. The roots can uptake water from the canopy soil, or those trees can uptake water from the canopy soil. And actually that water is not retained in that area. It's also redistributed to different organs of the tree. Now, our last question is how actually if that water is available at the canopy soil. So those plants with canopy soil, how they deal with drought stress conditions. If that could be extra water source that helps them to alleviate, or it could be an advantage to deal with water stress conditions. So for this experiment, we did 3 different experiments in different years. We used exactly the same treatments that we've been using for the other experiments. And then here, you can see here on the bottom, there is a panel with, on the y-axis is plant water content. And then on the x-axis is just time, or days of the experiment. And so we were expecting that actually the plants that were well watered, we're expecting that the plant water content doesn't change. And then for the completely dry plants, this plant water content over time will tend to decrease because they don't have any water. But the plants with the canopy soil, because they have water available at the canopy level, probably they're going to reach as low as the completely dry plants, probably not as good as the well water plants, but in between. And that's what we were expecting, if actually that canopy soil and that water from the canopy soil is helping them to deal with drought. So these are the experimental design. Again, all the plants were connected to, all the canopy soil were connected to IV bags, and we were very careful to avoid water reach the pots. So we did different type of measurements, physiological measurements. We did daily quick screening with fluorescence, net photosynthesis, stomata conductance, chlorophyll content, and also we measure hydraulic dynamics, so soil water tension and water potential. So the first that we saw so is actually so CS refers to canopy soil. How does canopy soil impact the soil moisture tension over time? And so what we were expecting, we used soil tensiometers, and then we were expecting that a plant that is well watered, the tension. So what actually this machine measured is actually the force of the water molecules attached to the soil particles. How strong is a water molecule attached to the soil particle? And so for a well-watered plant, we expect very low tension, but for a completely dry plant, we expect a higher tension, and for a DW plant, we expect probably lower. It depends on how actually that water works. So what we see, these were our results. So if you can see here, this is the y-axis is soil moisture tension in kilopascals, and then here on the x-axis are just the days of the experiment. And so if you notice here, the well-watered plants are maintained constant. But then when we saw the completely dry, that's what we actually expected, because they were completely dry, so the soil moisture tension decreased. And then for the DW plants, actually didn't decrease as much as the completely dry plants, which was very interesting here, since we didn't water the pots. Same and similar results we saw it on 2017, not as dramatic as 2015, but in 2018, actually, there were no differences between the completely dry plants and the plants with canopy soil. And so this was a very interesting result, because it made us think a lot about other different phenomena. It could be that over time, the plants with canopy soil were transpiring less, because probably they sense a signal from the pot, and so they were closer stomata, and they're probably transpiring less. And so it could be that, or it could be that actually the water from the canopy soil was redistributed to the soil, as we said that actually some of that water can be seen in the roots underground. Then we did measurements of water potential with a scalander pressure bomb. And again, higher water potential will be seen plants that were well watered, and we expect a low water potential for the plants that are completely dry. And so these were our results. So as you can see, the well watered plants maintain their water potential higher. And then we see here the completely dry plants. And then when we see the plants with the canopy soil, they are in between and actually closer to the well-watered plants compared to the completely dry. And 2017 is not as same, but actually we see a same pattern. So the plants with canopy soil are in between those two. And same thing we saw on 2018. So it seems that there is some effect of the canopy soil that is helping them to maintain their water potential. Now, if we tested chlorophyll fluorescence, that will give you, it's just a test to check actually if how the photosynthetic or the photosystems are working and how stressed are the plants. We expect a higher fluorescence for the well-watered plants and a lower fluorescence for the completely dry plants. And so again, we saw that the well-watered plants, the fluorescence is keep constant, and the plants that were dry, it decreased as we expected, because they were under very stressed conditions. They were completely dry, and the DW there didn't send, so it's in between both. It seems like they are not reaching the same stress as the completely dry plants. Same thing we saw in 2017, and same thing we saw on 2018, which means, again, that the canopy soil is doing some effect or is alleviating that stress conditions in the pot. Finally, we did photosynthetic rate with the liquor. Again, we are expecting higher photosynthesis for the well water plants, lower photosynthesis for the completely dry plants. And then what we see in this only 2 years is that actually, well, again, DW plants or they were in between both treatments, and same thing for 2018. So here they started at the beginning the same, but then again in the middle, they ended in the middle of the two treatments, well water and the plants with Canopy Soil. So this only confirms that actually there is an impact of the Canopy Soil for the plants that have it, and if there is an drought conditions, they could kind of alleviate that stress condition. So this is a photo of at the end of the experiment, and as you can see here, the completely dry plants, all their leaves are wilted. And then this is a well water plant, but this is actually a plant with the Canopy Soil. And as you can see here, they actually maintain their green, even though there are some leaves that actually were dying already, but it survived and it looks better than the completely dry plants. So yeah, then this also confirmed that the Canopy Soil is having certain impact and have some influence in helping them to alleviate the stress conditions. So if we go back to my main hypothesis that the Canopy Soil has an impact on hydraulic redistribution and plant performance, particularly under drought conditions, we saw that first, so this is just a tree and each system was connected to ivy bags. We saw first that actually these plants can develop canopy roots, and these roots are actually functional because they have the main structures or basic structures to be functional. We saw that actually those canopy roots, or that the tree actually can uptake water from the canopy soil with the dye experiment. We also saw that with the isotopic experiment and with the subflow sensors that actually there's enriched water in different organs of the tree. Even some of the trees show like enrichment in the roots underground. And also we saw with the subflow sensors some reversal flow. And finally, with the physiological performance, we can say that the canopy soil is working as a new water source or extra water source that is helping the plants to maintain their leaf water potential, that helps them to maintain their fluorescence, their photosynthesis, and their transpiration. And even though we have a very not conclusive about the soil moisture tension is not very clear. I guess it needs more experiments, but it could be that there's less transpiration, or it could be that also that water from the canopy soil can be redistributed downstream. So at the end, the canopy soil raises the water potential locally or at the canopy level, increasing that water potential gradient and producing possible reverse flow, allowing plants to maintain their plant performance. So this is the end, and then there are several things to do and still to do. I wanted a lot to do these experiments in the field, but it wasn't possible by the time. The permits were not possible, but it will be very good to, because most of the experiments were done in the greenhouse, so it will be really good to see if actually that water is redistributed within the tree in the field, and with real canopy roots. Also, it will be good to see if actually that root, how much of that water is absorbed by the canopy roots and how much of that water is absorbed by just the bark. What are actually the environmental conditions and signals that induce the formation of canopy roots? What is the hydraulic conductivity and capacitance across the tree body? What if actually probably those canopy roots are not only uptaking water, of course, they're probably uptaking nutrients. So what kind of nutrients are uptaking and how much of those nutrients are from the canopy soil? And also how it will be affected, the canopy soil, with climate change. It will be interesting to see. So with that, I finished my presentation. Thank you for everything. These are just pictures of the Pacific Northwest Forest. And then just acknowledgements of my advisors. This is Liz van Volkenburgh and Soo-Hyung Kim, and these were my two labs, the Grow Lab and then the Plant Ecophysiology Lab at the University of Washington. Also, this project was funded by the University of Washington, NSF, the National Science Foundation and the University of Utah. And then thanks to my committee members and Nalini Carney, that is the person that actually discovered the Canopy Roots 20 years ago. Actually, it's very recent. And you can find her as the Queen of Canopy, and you can find a lot of YouTube videos about her. And also thanks to Todd Dawson that helped me to analyze all the isotope measurements. And that's it for today. And also my parents that were able to help me in the greenhouse. And here they are here working, and they help me to collect measurements. That's it. Thank you for everything. And if you have questions, just let me know.
So, hello, Johanna. Great presentation.
Thank you.
If you watch the comments here that everyone made, a lot of them are talking that it was a nice presentation, so I don't have to say it. So we are a bit short on time, so maybe we cannot reach all the questions. So we try to group some. So
Yes.
I will ask you 2 questions or more at once. Okay. So are you listening to me? Your
Yes.
image is freeze. Okay. So the first is from Gabriela. She's loving the subject, very interesting, and she asks if there are similar studies with cannabis soil in Brazil. Do you know anyone?
I don't know, but I would like to. I don't know. I know there are a lot of studies of canopy soil actually right now in Costa Rica. They got a big project there, but it would be very interesting. I mean, I haven't seen any project like in South America by itself. No, there are no projects about canopy soil. It's sad, but it's very interesting. Yeah.
So someone asked if the Atlantic forest has the same potential in terms of canopy soils.
Sorry, the what?
The Atlantic forest or the Amazonia, the tropical forests overall, if they could have a similar behavior.
Yes, a lot. Soil. Yes, it actually has it. And so that's also because in Costa Rica is also tropical forest. They have seen a lot of canopy soil and a lot of canopy roots there. It's just that it's been very difficult to access to the canopy. But now there are a lot of people doing research now that we have more access. But yeah, there are a lot.
Is there any information about the diversity of species of the canopy soil in different tree species in the same region?
So actually, yeah, it depends of the species. So not all the trees can develop canopy soil, or they can develop canopy soil, some of the trees, but not canopy roots. So it's different. So some of the species have that ability to develop canopy roots. And we don't know, and that's why I put in my feature directions, that we don't know exactly what are the specific signals that made some trees develop canopy roots and some of them not. It seems that it's a mix of a group of different factors. It could be humidity, hormonal, light conditions. There are several factors that will affect the ability for those trees. But yeah, there's it's a species-specific condition. Yeah.
Yeah. Very nice. So very, very interesting relationships. Would it be right to say that the cannabis soil behind helping in this water redistribution could act also as a water reservoir?
Yes, exactly. That's what I was thinking. And yeah, it's actually working as a water reservoir or potential extra additional water in the canopy. And so we were thinking that probably if the soil underground that can get drier by climate change, because now we talk about warming. It could be probably that those mosses or those canopy soils can retain a lot of water or maintain water that helps them to survive or to alleviate those conditions. So yeah, it can work as reservoirs.
Well, this one is in English, so you can.
When we talk
see
about
for
forest
yourself.
succession, the large presence of epiphyte is a characteristic of all growth forests, as well as the presence of taller trees.
It continues, yeah.
We say that there is a context of facilitation there, whether this canopy soil and the presence of epiphytes help. Yes, I agree. I think so. I think it could be like, yeah, facilitation. Yeah. So actually, there's other way for taller trees, and I show you sequoia, but there are some trees also at the Pacific, not the Pacific Northwest, but at the southwest in California, that those trees don't have canopy soil, but those trees also have their leaves can absorb water from the fog water or from the atmosphere water. So there are some adaptations that taller trees have besides canopy soil that also helps them to survive and to avoid that resistance to move water from the soil from the bottom to the top. So yeah, there are like a lot of adaptations. Could be the canopy soil, or could be that the leaves are able to uptake water.
Okay. Yeah, good luck with that one.
Could there be a relationship between the evolutionary emergence of epiphytes and canopy soil? Relationship between the evolutionary emergence. I guess so. I evolutionary. I mean, I guess where there are epiphytes, simultaneously when they decompose, they accumulate that organic matter and they become canopy soil. So I guess yes, they evolve simultaneously. Could be. Not sure. Yeah, but I mean, it's all that organic matter that decompose and accumulates over the branch. So yeah, eventually those epiphytes are gonna die, and so they're gonna be accumulated. Yeah, over there. So yeah, they're part of the canopy soil.
Yeah. I was thinking about maybe competition between the epiphytes and the host tree for this canopy soil for the
Yes,
resources.
I discussed a lot about that. So there must be and that's why we wanted to also be able to track how much of that water is available actually for the host tree and how much can uptake and compete with all the epiphytes. But I guess, as I said, and as I mentioned before, it probably the trees are those canopy soils are not the main source of water. Of course, it will be the soil. So probably they will be more just in case they need it. Probably they will use that water source, but not always if they don't need it. If there's more available water in the soil underground,
So
I feel.
it's not a very strong competition.
Yeah, I think.
Maybe. Um, well, go for it.
If I know they use, I mean, they use canopy soil. Not, I haven't heard about some applications of the canopy soil. Um, no, no, I haven't. I haven't heard. I don't know. Just for decoration, I have seen it, but not for like applications for agriculture. Yeah, I haven't seen that.
Okay. I will make a question that is from myself and from Maria Alice as well. So, if you think, actually, it's mostly from Maria Alice, then I copied it from part of it. She asked if there are fungi in this canopy soil that help the plants, and I take a little broader to microorganisms in general.
Yes, they have found mycorrhiza associated to those canopy roots. And also they have found not only mycorrhiza endophytes, also they have found. So they are like, nitrogen fixing endophytes and mycorrhiza associated to the canopy roots. Just very cool. Yeah.
Nice. Do we have a little more time? I will make it. I make my question here. In your results on the on the soil water potential, you saw that the presence, sometimes in some years, the presence of the canopy soil increased the water potential of the soil down there.
Yes.
Could it be that the plant is actually watering the soil?
That's what I was thinking. So we don't know, and that's why it was so inconclusive, those results. If actually if it's the plant that is watering the soil by itself, which it will be very weird, but because of the difference of water potential gradients, it makes sense. But we don't know. We need to.
I was just thinking because there is a whole microbiota there in the roots, and maybe the plant is trying to keep them alive as well in a bad condition. So
I thought the same. Yeah, it could be just a hydraulic redistribution in the soil, and
around the roots, so it attracts more microorganisms from elsewhere.
Yeah, it would be a good experiment to do. There are so many things to do about
really, really interesting.
Yeah. Yes.
So I don't know if anyone has another question. Have we skipped any one of them? I don't know about the time either. Maria Alice, give us a sign. Okay, she's saying that we can end here. So thank you very much, Johanna.
Thank you very much.
It was a really nice talk. Everyone enjoyed here. Lots of questions, lots of compliments.
Thank you. If you have questions, and you can also email me if you have more questions later. I don't know if you can share my email. It's fine.
Okay, so if you put here in the private chat, then I can send it to everyone that's on this discipline. Yeah.
Yeah.
Okay.
So if you have more questions later, we can discuss.
Yeah, I do actually. We need to take a beer and ask some questions.
Yeah.
Okay, so thank you, and see you another time in the future.
Thank you. It was nice. Thank you. Bye. Thank you, everybody.